Good morning. I did my bioinformatics project on frontotemporal dementia, and the reason why I picked this was because this disease is very similar to Alzheimer's, with the aggregates developing in the brain, basically suffocating the neurons, causing plaques to form, which then lead to, dementia, memory loss, personality changes, and we're seeing more of it within our population. So I really wanted to investigate this and learn more. The basis of this project is to find out the model organism at which I would do research, and figuring out kind of what organism I would feel would be beneficial to investigate the development of this disease. So, no. So when we're looking at FTD, or frontotemporal dementia, there are certain diseases that are associated with this, and one of them is Pick's disease. You have different forms of FTD, and they are based on what chromosome we are actually dealing with. In the evolution of FTD article, they talked about 3 different types of mutations. One of them is prevalent on chromosome 19, one of them is chromosome 9, and the other one is chromosome 3. The majority of the population that suffer from this disease are on chromosome 17. So this slide shows kind of the progression of the disease. So we have Pick's disease, and then right here, the MAPT becomes very important with chromosome 17 and the mutations that are involved in that. And so we also see MAPT over here. And you can tell that when we're dealing with brain diseases, we're also dealing with Parkinson's, we're dealing with Alzheimer's, we're dealing with dementia, we're dealing with a lot of diseases that show up later in life, and sometimes that show up early in life. So what this disease is since I focused on chromosome 17, we are going to be talking about tau aggregates. And so tau is a protein that stabilizes the microtubules, as seen here in this image. So with our microtubules, tau will come in and stabilize the development, making sure that the structure stays consistent, the structure is rigid, and there's no misfolding within those microtubules. But when tau has a mutation within it, so this protein folds incorrectly, it forms defective microtubules. And so these aggregates, the tau aggregates, or in this case they call them tangles, cannot allow the microtubules to be stable. And so it basically suffocates the neurons because the tangles don't allow the chemical interaction, decreasing the function of the neurons. So here they isolated these mutations in as I stated before, in chromosome 17. Here in this study, they looked at chromosome 17 and where these mutations actually happen. And so we are looking at the PGRN mutations within specific families that show FTD. And they show you basically the wild type and what we would see with a normal development of the protein of tau. And then it goes through and shows all of the different types of deletions and substitutions that can happen that cause this protein to misfold, causing the aggregates. And then they really focused on the MAPT and the PGRN within chromosome 17. And this disease is autosomal dominant. From all the articles that I've been reading, both with chromosome 9, chromosome 17, and chromosome 3, they see this progression through the generations. And they stated that it's autosomal dominant within the families. Some research has shown that there are certain populations or ethnic populations that are more prone to having FTD. There were 2 studies done focusing on China and showing that there's a heavy prevalence of FTD within their population. So here they are linking it to the MAP2 tau. The really interesting thing, and taking into consideration this specific protein, is that there are so many isoforms that can develop with this protein. So when looking at a model organism, I really want to make sure that this model organism has a system in place to where I can manipulate these isoforms and investigate which ones are more important, where we see deletions within these exons, so that I have it there in order to investigate tau and MAP2 relations. So here is a table that is showing you the different types of isoforms that both of these have. So when looking at tau on chromosome 17, you see that it actually has 4 isoforms. And so the 4R2N, the 3R, those are related to the repeating sections within there, and within the protein, And so these are, looking at what's repeating within the isoform. And so when I was doing my investigation into all of the genetics of all of the different organisms, it was really important that the protein that was established, or the homology that was within those organisms, had the ability to have these different isoforms. Because in the research that I've been doing on this disease, there are certain isoforms that are more prevalent within FTD cases, and also PIC, and also Alzheimer's. And so I want to have a mechanism already in place to allow me the ability to do the manipulation and to see how it affects the health of the organism that I'm investigating. So again, this is another illustration of the isoforms that can develop, and looking at where we're actually seeing these repeated sections. You can also see where on the 4R you have a conservation within the repeating sections. And so these are really important with the development of this protein and the proper folding and dealing with the phosphorylation of the protein, because when these isoforms are not in correct sequence, or there's a mutation, or there's a change of one of the bases, it can cause this protein to be phosphorylated, and it then causes it not to fold correctly, which then inhibits its ability to stabilize the microtubules. So I went through, in all of the databases that I went through AMID, GenBank, National Institute of Health, NCBI. So when going through all of these, I was looking at kind of what are the introns and exons that are available within the different proteins. So this is chromosome 12. And looking at, kind of what is similar, what is not similar. Are there homologies? Are there systems already in place so that I could manipulate these systems with this organism in order to be able to do an investigation on this disease? And so this is the data that I collected for zebrafish. Zebrafish were not, are a good candidate for me when dealing with tau and tau aggregates. There's not a lot of homology within the protein, and so there's really not a lot of this isoform within the protein. And so I went through homogene and looked at kind of what is conserved with this chromosome 17. And so it looked at the protein tau and kind of gave me what are the organisms that really have homologies within this tau protein. And so it gives you the structures over here and shows you basically the comparison of the sequences and the sizes and looking at what are conserved, what's not conserved within them. And so when going through this list, obviously these are organisms. I mean, mouse models are always with this type of investigation into diseases is used a lot. In all of my readings, mouse models have been used for decades when dealing with brain diseases. So obviously it shows that mouse is a very good candidate for an investigation into this disease. And so this is chromosome 17 for humans. And so I used this in order to compare it to the other organisms that I was investigating as potential candidates. So here's the protein coding region. So I used this protein coding region in order to go into other databases to figure out which candidates would be the best to do an investigation on. And so again, I'm just showing you basically, hey, zebrafish, not a good candidate. Now this was really cool. So this is a nematode, and so in the databases there was not a lot of information on nematodes and fruit flies, but in the research and the reading of all of the articles and everything that I find, they are finding different sets of genes that are like tau and have homologies to tau. And so I read two articles, one of them dealing with fruit flies and the other one dealing with C. elegans. And it showed that it has this protein that acts like tau, has isoforms like tau, so the exons and introns, and very similar pathways. And so in this article, they were focused on C. elegans and showing basically the homologies through this tau-like protein. And I found this really interesting because if you can move to C. elegans, then you're dealing with something that, has many generations in a quick amount of time. You can do a lot more, and you can see a lot of the interaction within the muscle structures. And so it would be really interesting if this tau-like protein that they're finding in C. elegans would be usable in research. And so this protein was PTL 1A, And so it went, they did a lot of investigation kind of comparing it to tau, and as this diagram shows, there's a lot of similarities between the two, and also with MAP2 and MAP4. But the really cool thing is when reading this article, it really started to investigate the isoforms, because the isoforms are kind of key to the development of this disease. And C. elegans, this PLT, actually has the ability of creating isoforms, and I believe that PLT, or PTL, I'm sorry, 1A has the ability to make 3 isoforms naturally, if I remember the article correctly. And then also in a different one, they were looking at fruit fly. And so they found a homology within the fruit fly that is tau-like. And so here we're comparing C. elegans PTL1A to humans, to fruit flies, and looking at what are conserved, what's not conserved within this protein. And so fruit flies also have a conserved unit. That is very important. So this MTBD repeat. Those are the repeats that are like the 4R, the 3R repeats. And so it's really important to be able to show that we have these repeated sections within the protein, so that we can do investigation when, these repeats are phosphorylated, or they're not phosphorylated correctly, or there's not interaction with the MAPT1 or the MAPT2 or the 4, in order to create the proper folding within the protein. And so here it shows that C. elegans and fruit flies dystrophia are or do have these repeated sections in tau homologies. The really good thing would be is that, both these organisms, C. elegans and dystrophia, are quick generations. So you can have them in the lab, and you can look at multiple generations over a long period of time and see how this affects through crossbreeding and creating homozygous pairing of certain types of mutant strands. And it would be beneficial for the research, because then you can create different types of mutant strands in order to investigate kind of like this evolutionary aspect of FTD, and why certain ethnic groups are more prone to having this develop within their populations. So here it's kind of showing you the layout, with tau, mouse, human, we have very similar homologies genetically. But then down here, they're starting to show that this PTL1A and the CG31057 are also a part of this isoform tau-like homologies, and are viable within the research community. So again, here's the same thing. It's just showing you that within here are microtubule formations and the interaction of the binding unit, and showing you the conservation and where we would have all of the interactions within tau and the isoforms. So these are all my articles that I read. I read a lot more, but these are the ones that I kind of pulled for this presentation. When looking at FTD, I think the beneficial, if I were to do this research, I would use C. elegans. And the reason why I would use C. elegans is because in the research that I've been doing, on this organism, that the protein that they're finding, this PTL1A, has the ability to create these isoforms, and those isoforms are very similar to the tau isoforms that are seen in humans. And so the genomic sequencing of the PTL and also tau are very similar, and they're conserved. And when we're looking at the different forms, they're seeing that these are very similar to each other. Then with the folding of PTL, so the research in a couple of the articles showed that when we're dealing with the folding and the misfolding and creating the aggregates in C. elegans, the aggregates, you also have the insoluble and soluble, and they cause you can see the aggregates within C. elegans. And I think that pathway would be very important to investigate and really look at these isoforms and how they interact with the phosphorylation and the folding, so that you can do a quick investigation instead of waiting generations and generations with mice. With C. elegans, you can do very quick generational crosses, and especially with the development of these isoforms and the development of the disease. Now, the other really cool thing, I've been kind of on the fence about fruit flies. In the research associated with fruit flies as a model, when you're looking at it, I think some of the problems, and the reason why I picked the nematode over the fruit fly is because in some of my readings with the fruit fly models and looking at the gene that has isoforms, the isoforms aren't as prevalent within the fruit fly. And then also there was an article about fruit fly and how we're dealing with the vision system in the larval state on these aggregates. And so while reading through that, the article was, there seemed to be a lot of holes within the argument of actually this homology within fruit flies. And I wasn't as positive about using fruit fly as my model organism because I felt the C. elegans protein was genomically more similar to tau and the isoforms that we see with humans. The fruit fly, it had similarities, but the function within the larval state was different. And so that tells me that there are other things working on this protein that we don't see in our human model. While in C. elegans, in the development of the protein, it's a very similar pathway, with this PTL and how it folds and the interaction with other proteins in order to create the folding and the interaction between some of the other genes. I felt that C. elegans would be a better model because the system to develop the protein was extremely similar to the human development of tau and its interaction with the microfilaments. So that's my presentation. Again, this is all about FTD, and I hope you have a wonderful day.